We have covered a lot of class 1 fusion proteins. Now those are going to be proteins similar to the HIV envelope protein and the SARS coronavirus spike protein and the influenza virus HA hemagglutinin protein. But there are two other general classes of fusion proteins out there, and then there are quite a number of proteins that seem to do the job but don't fall into one of these classes. So the classes are named because we have multiple different viruses of different types that seem to be using the same general mechanism. So let's talk about class 2 and class 3 viral fusion proteins. This will be helpful if you're doing the compare and contrast mechanisms of entry question. So examples of class 2 fusion proteins would be what you find on Flaviviridae and what you find on alpha viruses, which are Togaviridae. These proteins are going to look the same, do kind of the same thing. There are some other viruses. I believe a lot of the Bunya viruses would also have class 2 proteins. But let's focus on this one, which is Dengue virus fusion protein. So here you go. These proteins are going to start out very flat to the surface, and there's a very nice video that you can watch down here that shows that. They look just like a volleyball, same sort of pattern on the surface and everything. And the glycoproteins, which are shown here, are going to be folded down and they're going to be paired, so there are going to be two of them attached to each other. That is the structure that's actually going to bind to the cell. And so once it binds to the cell and gets internalized, gets pulled in through the endosome, then we're going to have acidification of the endosome, like always. And when that happens, these two proteins are going to pivot and they're going to stand up. And a third protein, same type, but from one of the other pairs, is going to come over and join it. And so you're going to make a thing called a non-crystallographic trimer. It's just like a sort of a wonky three proteins leaning on each other kind of thing. It's not symmetrical the way a class 1 viral membrane fusion protein would be symmetrical. So you make this asymmetrical trimer of three proteins, and that's going to happen after acidification. And so at that point, see the yellow domain here? That's domain 2. Now it starts out tucked down, and at the very tip of domain 2 is the region that we think is the fusion peptide. There's still some controversy here, and it may turn out that this has to be revised. This may not be the fusion peptide, it may be a region involved in fusion, but right now it looks like it's in the right place and has the right properties, so that's probably it. And it is an important part of the way this protein works. So the tip of domain 2 is going to stand up, and that is basically exposing the fusion protein. So we don't need a furin cleavage, we don't need a tempress 2 cleavage. It's just going to be dependent on binding to get you inside the cell, and then acidification inside of that endosome, and that is what is going to cause the conformational change, and the change from a dimer to a trimer. Like that. Okay, once we've done that, the domain 2 is going to be poking into the membrane, roughly as shown here. They've kind of unfolded the thing. And then here again, we have to interpolate a little bit because we can sometimes get structures before and after this process, but there's not really a good way to get a structural read on what happens in the process. You can model it, and the models seem to make sense. For now, that's probably as close as we can get. So you're going to have domain 2 up here hooked into the cell's membrane. So imagine that is buried in a cell membrane. And down here at the bottom, you're going to have the transmembranes of the actual class 2 fusion protein. So the protein is called the E or envelope protein here. And yeah, that's the same name given to the tiny little thing that helps coronaviruses leave the cell. Which just goes to show it's kind of a boring, ordinary name. But yeah, Flavivirus E proteins are well known. Anyhow, so we go from there, and then this is going to pivot like a hinge. So domain 1 is going to be the hinge region, and it's going to go from stuck into the membrane, and it's going to, boom, it's going to pull back and snap the host membrane down into the viral membrane. This is how it is understood to work. And domain 3 is there to sort of hold things together while these guys are pivoting. They're basically going from domain 2 on the top and 1 on the bottom to 1 on the top and 2 on the bottom. And 3 is going to get pulled up where 1 is while that happens. So that is the mechanism. My super duper shorthand here is that we're going to go from a dimer, we're going to bind the receptor, we're going to get internalized. And then after internalization, which means like entering the endosome, that's going to acidify. The acidification is going to trigger the dimer to trimer switch, and we're going to stand up tall now and expose our domain 2s with that sort of fusion peptide-like thing on them. Domain 1 is going to act like a hinge, and weirdly, yeah, notice how the domains are labeled. They're kind of backwards. They're labeled the way that makes sense on the structure. But yeah, domain 1 is the first part of the protein. That ends up outside, and there's domain 2, then there's 3, and then there's 4 down there. Yeah, I don't know. I tend to think of it in the opposite order, but it works. I guess this looks like a class 1 fusion protein if we number them this way. So yeah, there you go. That's the idea. And then that'll lead to hemifusion and then fusion. And this video shows it very nicely. It's a molecular simulation, so it's not an actual photograph of the virus fusing, but it's about as close as we're going to get for now until there are major advances in structural biology. This thing at the top, that's the SARS coronavirus class 1 viral fusion protein doing its little exercises. Yeah, doing some little chin-ups there. It's going from pre-fusion to post-fusion. All right. So that's class 2. Dimer to trimer, like a little soccer ball that then turns into a spiky soccer ball. All right. Class 3. Yeah. Class 3 is in session. Okay. The most famous protein from class 3 is going to be the GB protein. The little g stands for glycoprotein. So it's glycoprotein B, and it's got lots of friends. It's going to play with a glycoprotein D, which is GD. And there's one that's called GH, and it's also been called GL, so people now just call it GHGL. Yeah, it's like you can't decide on a name, give it both names, and move on. The H was for high molecular weight. The L, I think, was for large. I believe that's correct, or it may be that you can cleave it into 2 pieces. I don't know the herpes virus literature quite as well, yeah, for the origin story of this particular villain. But yeah, this is herpes simplex. This is the one that causes cold sores. You get the same type of protein on baculoviruses. That's an insect virus that's very useful in biotechnology because you can use it to grow tons and tons of protein in insect cells. And insect cells are a lot closer to mammalian cells than yeast or E. coli, for example. So yeah, it's nice. All right. Other ones, vesicular stomatitis virus. It also has a class 3 membrane fusion protein, and there are a couple more out there. So how does it work? Well, our virus is going to bind, and it actually uses several different proteins that are going to work together. So GD is the main receptor binding protein. It's going to do the attachment. And the main receptor is HVEM, and the co-receptor is nectin-1. And so GD is going to bind those, and that is going to bring in the GHGL. So GD holds on to something, and then GHGL comes over onto the side and it's like, oh yeah, me too. I was totally doing this. All right, so now that we've got that, GD is going to stay attached, so it's doing the job of the S1 region of spike, or the GP120 region of HIV, or the HA1 region of hemagglutinin, if any of those mental hooks are in your brain and we can hang it there. Yeah. So it's just like having that, but it's a separate protein and it's got its own transmembrane. So instead of it being part of the fusion protein at the top that gets pulled away, it's just already pulled away, but right beside the others, doing the same job. Yeah, it doesn't look the same, it just does the same job. That's why this is a class 3 and not some kind of mutant, weird class 1 fusion protein. Okay, then we get to the structural gap. So we know that GHGL will interact with GB, and GB is the actual class 3 fusion protein. So GD and GHGL, those are going to be attachment factors, and GHGL is like a cofactor. It's like the middleman. It's going to sort of pass the buck, yeah. Like, okay, GD's doing its job, now GB, it's time for you to go. So it's going to recruit GB, which is floating somewhere nearby, but once we bound the receptor, then GHGL can actually bind to GB and bring it in to the correct site. So we just got these GBs floating around, and then they get, boom, brought in together, just like that. What you see here is the post-fusion structure of GB. Now, we know from vesicular stomatitis virus, we have the pre-fusion and the post-fusion structures. It's just that the visuals aren't quite as nice and thorough, so we're going with this one. But what those visuals show is that when it's pre-fusion, this big green domain, which is domain 2 mostly, is going to be folded down. It's sort of tucked down on the outside, just like that, of the protein. And then when we fusion activate, which is going to be acid dependent, it's going to stand up. And so the interesting thing here is that the fusion peptide is believed to be in GB, but it's going to work in the opposite way. So GB is going to start out embedded in its own membrane, and it's going to be binding lipids from there. And the fusion, motion, if you want to think of it that way, is going to be bringing lipids from the virus membrane up. We're actually pulling the virus membrane up and merging it with the host membrane. So instead of conceptually, I think of class 1 and class 2 proteins as pulling the host membrane down to the viral membrane. Although there's, I think they all meet in the middle, really. But yeah, this one starts out embedded in its own membrane, and it pulls that up and then merges it with the host membrane. So there's your class 3 membrane fusion protein. And it's this part up here, domain 2 of GB, that's going to do that. Now the other neat property about class 3 membrane fusion proteins, they don't seem to require a receptor themselves. You can express these things, and if they get close enough and they get acidified, they will stand up and fuse all by themselves. So it's like a fusion protein that's independent of binding. And so the things that make it dependent on binding that help keep this thing alive are GHGL, which is coordinating, and GD, which is actually doing the binding. So I think that's kind of interesting. The other interesting thing is that for class 1 viral fusion proteins, remember all those conformational changes where they go up and they hook the host membrane, and then they're going to crash those helices together and bring it back down? That's a one-time deal. Once those spike proteins or HAs or envelope proteins are acidified, they go through an irreversible conformational change. They can do this once, unlike the little animated GIF above, which is doing it over and over and over again. Yeah, that doesn't happen in the real world. So class 1s only get one shot. Class 2 fusion proteins, they can do this part where they go from dimers to trimers, dimer, trimer, dimer, trimer. They can do that as many times as you want. Every time it gets acidic, they'll stand up. Every time, if they don't fuse, they'll go back down. But once they've hooked a membrane and unfolded like that, sort of done that hinge thing to bring the host membrane down to the virus membrane, From that point, it is irreversible. As far as we know, the full process of GB is reversible because it starts out in its own membrane, and if it reaches up and it can't grab a host membrane, that's cool. It can just go right back down, if the acidity goes away. There are histidines, of course, around this hinge region that are going to assist it in sensing pH, sensing it's inside the cell. And that's going to be the same over here. There are histidines along the dimer interface, if I remember correctly. And those are going to get protonated, and they're going to go from happy and hydrophobic to real positive, and they're just going to blow this thing apart. And so, the happy little dimer will get turned into a very angry trimer. Is the way I think of it. There we go. So those are class 2 and class 3 viral membrane fusion proteins. Hope that helps, and hope that helps them make a little more sense. Both of these are from the same paper, so if you want to read about those a little more, the link is in this paper, or rather will be as soon as I go and edit it. Thanks very much.